Dear ladies and gentlemen, we are very happy to welcome Dr. Amy Pyle as our guest today. Um, Amy, could you tell more about the research you are doing and about your everyday activities, as well as what is your biggest goal and what is the mission essentially of that what you are doing?
Sure. If it's okay, I would like to start with just a little bit of background and how I got to where I am. So I started out in a traditional biomedical research track, and I earned my PhD at Vanderbilt University in the Department of Pathology. And after I graduated, I was looking around for opportunities and I was looking at postdocs. And then I stumbled into the career of clinical chemistry and pursued a clinical chemistry fellowship. And I'm now practicing as a board certified clinical chemist. And so what clinical chemistry is it's one part of a clinical laboratory. So anytime somebody's in the hospital or goes to the doctor and they get their blood drawn, it's sent to a laboratory like ours. So this is kind of it's a really busy, large laboratory, largely operated by medical technologists. I'm one of the directors. There's many different areas, so I'm in charge of chemistry and special chemistry. One of my colleagues, who's also a clinical chemist, is in charge of biochemical genetics and metals testing. There's also hematology and coagulation. There's genetic testing and microbiology and anatomic pathology. So it's very broad in scope. Specifically, what I do in chemistry is measuring anything that is dissolved in blood. So that can be something like sodium or potassium. It could be cholesterol and lipids or hormones like thyroid hormone or parathyroid hormone. So we do across all of chemistry, probably about 250 different tests or assays. And we do, thousands of samples a day in our hospital's laboratory. So most of my job is spent overseeing kind of the technical and scientific aspects of this laboratory. Unlike a research lab, everything we're doing is very, very clearly spelled out, very proceduralized and standardized. So we're not, taking patient samples and adding a little bit of this or a little bit of that and seeing what comes out. Everything is very routine and somewhat automated. And so that we're producing really consistent results. When our quality control measures start to show that there's drift in the consistency of those results, I usually step in and help with troubleshooting. And there's multiple variables and factors that we have to kind of rule out. It's traditional troubleshooting approaches. And so a lot of my day is spent just working on that sort of thing. And then working with clinicians and answering questions that they may have about their results or if, how they order the right tests, stuff like that. My research specifically, I deal with kind of two areas. So I have a pretty long-standing collaboration with some of our pediatric endocrinologists here at my institution. And we have been using laboratory results generated from my lab and the clinical picture and integrating those to look at optimizing diagnostic measures for specifically, right now we're looking at cortisol insufficiency and growth hormone insufficiency. So really looking for kind of evidence-based practice, because a lot of times what people are using as diagnostic cutoffs for various diseases are based on old methodology, very old studies. And so there's a need for ongoing research and maintaining kind of up-to-date cutoffs like that. So that's one chunk of what I do. And then another chunk of my research is spent working on kind of optimizing sample collection, sample stability, sample storage for the samples that we receive in our laboratory. So one area in particular that I've been working in for several years is the best way to collect urine from little babies. So really anybody that's not toilet trained. So somebody wearing a diaper can't void on command into a cup. And so there's a lot of things that people will do to try and collect a urine sample from a baby. And they all have advantages and disadvantages. A lot of them are very tricky. So probably the most common ideal one is to take a plastic bag with an adhesive around the outside opening of the bag, and you adhere it around the genitals of the baby and then put the diaper on and wait for them to urinate. It is incredibly challenging to actually get a good sample. And so one of the things that a lot of people do in practice is to just put cotton balls in a diaper and then just squeeze the urine, express the urine back out of those cotton balls. And as you could imagine, this obviously the urine sample that's obtained is far from a clean catch, and it's going to have the microbial flora of whatever's in that diaper, but also whatever chemicals are in the diaper or in the cotton ball can interfere potentially with testing the urine sample for urine chemistries. And so there have been numerous studies over the years looking at kind of a few aspects of this kind of collection, but nothing really comprehensive. So we've done some comprehensive studies looking at kind of all the common urine chemistry analytes and some of the common urinalysis studies, and shown which are most affected and by that kind of collection. So we're doing that sort of thing, and numerous other variations on sample collection and stability.
Okay, so essentially, to summarize, you have two lines of research. The first one, it is about the analytics, right? This is analytical chemistry, how to detect specific molecule, whether this is a hormone, whether this is some other substance in any body fluid or in any specimen, right? In the human body, probably also in the animal. And the second axis is how can one collect the samples, right? From the humans. Looking on the first line of the research, how often do you find it necessary to review the thresholds of those specific molecules that were set a long time ago in the guidelines? As I understand, they were set pretty long time ago, but it evolves with the practice. What are your points? What is your beacon, or as those red flags where you say, okay, from this moment we believe that it is the most optimal to review the level of this molecule, that it can correlate with other specific outcome or some hint to a disease and so on and so forth?
Yeah, that's a really good question. So unfortunately, I would love to tell you that we have a really good standardized method for identifying which analytes we want to review, but usually it's much more practical. It's the ones where clinicians have a concern about our thresholds, or honestly, the main research projects that we're doing are kind of the ones where we have the best access to data, and where we have a fairly large referral service for growth hormone deficiency at our institution. And so we have a lot of test results to review for that. But just more generally, I think this is a great topic to delve into, is the issue of normal ranges for our tests. So how do we know that, say, a blood serum sodium result is normal or not, right? Every laboratory has to have normal ranges or reference intervals. And so reference intervals are really tricky because you need to have it to be specific for your laboratory, for the population you serve, and for the method that you use. And unfortunately, for a lot of assays, there's lack of standardization across the different methodologies. So for example, for a lot of our hormone assays, we run them on a platform called the Abbott Architect. And so that manufacturer these are all generally immunoassays, and they're using their particular antibodies that they have generated to recognize the analyte of choice. And the way that it picks up the analyte of choice and cross-reacts with similar structures is going to be different for that method versus another manufacturer's method. And so, a result of 10 by one manufacturer may, from the same patient, you may actually get like a 15 or an 8, depending on what method you use. And so you have to compensate for that using different reference intervals. Ideally, reference intervals would be established by taking 120 normal donors that have been screened to really be considered normal and healthy, and running their blood for that particular analyte, and you take the central 95%. What that means, though, is that there's 2.5% of people that are going to be healthy on the low end, or they're outside of your normal range, and 2.5% that are healthy on the high end of your range. So you're always going to have some patients that are truly healthy but falling outside of the normal range. And then, for some tests, you need to split these up and do the same thing just for males or just for females. And then it gets really complicated in pediatrics, which is the field I work in. And so there are many tests or analytes that change significantly throughout the growth and development. So one example would be alkaline phosphatase. This is the amount in circulation generally reflects bone growth, and it's high early on, and then it rises again during puberty, and it's higher usually for males than for females. And so to accurately do a reference interval study, you have to collect 120 males and 120 females from each age group. And so doing that is nearly impossible because it's very hard to get normal, healthy children to donate blood. And so there's a lot of data analytics that we're currently relying on to generate our reference intervals. And so there's a lot of research from my colleagues that's going on in that field.
Sure, thank you so much for sharing this information. Looking on your discoveries, right, those that you have done in both axes of your research, in the analytics as well as in the method establishment, what had been your biggest achievements? What are essentially the brightest moments of your discoveries?
Wow, that's I don't really think about my own research like that. It's a great question. Well, so
maybe just something astonishing, right? That information.
I would love to say I had a eureka moment. There's not been too many things that are like that, but I mean, what I can say is there have been a couple of papers that have been fairly high impact. So I was middle author on a paper that was talking about establishing pediatric reference intervals, what I was just talking about, and the challenges of that. And that was published in a pretty high impact journal and had an editorial. So I think that was really well received. And hopefully, it's not just clinical laboratorians that need to know how to establish reference intervals. Anybody who's interpreting a lab result needs to understand where those reference intervals came from and understand kind of the gray area that goes along with that. So I'm really excited that is picking up a lot of attention in the medical field. And then also some of the work that we've done on growth hormone deficiency was just featured in some of the best publications on pediatric endocrinology. So a pretty small, narrow subset of papers, but I think that again has been really well received because again, everybody's using diagnostic thresholds and cutoffs and ranges that are based on very old papers, or we don't even know where they're from anymore. And so having really legitimate data behind some of these numbers is really what people need. So I think that's been well received.
And so this is an interesting information. So essentially what you say that these thresholds, they vary from one research laboratory to another, or let's say it depends on the experience of the doctor. So there is no really that hard measure what is norm and what it's supposed to be. Or?
So let me give you one specific example. So there is a test called an ACTH stimulation test. This is for somebody who has suspected hypofunction of their adrenal glands. So the adrenal glands make a couple of things, but one is cortisol. And cortisol does a lot of things in your body. And if you don't have enough, it has wide-reaching effects. And so we need to test for that. If you just measure cortisol by itself throughout the day, it changes and it's hard to know what you're looking at with a single result. So what we do instead is you draw a baseline blood sample and then you give a stimulant. And that stimulant should, if everything is working normally, cause cortisol release from the adrenals, and you have an increase in circulating cortisol. And I think I initially traced back the publication to the 60s or 70s. A bunch of studies were done looking at a normal response to an ACTH stimulation, and they determined that the cutoff for cortisol was 18.5 micrograms per deciliter. And so for decades, people have been using 18.5 as the cutoff for that. The problem is that I have no idea what methodology was being used 40 years ago for measuring cortisol, and I don't know how those results compare to what we're doing currently. But I do know that even within all of the clinical laboratories and the major manufacturers, there's variation in how those results compare. And so we really need to establish our own thresholds for a normal response. And again, I can't go out and collect a bunch of, especially do this kind of stimulation test with multiple blood collections on children. So we're looking at the literature, we're looking at what other people have done, doing multiple comparisons between different laboratories and different methods to establish what the cutoff would be for our laboratory, which we eventually settled on as 15.5. And in doing so, we really changed the diagnosis of ACTH or of cortisol deficiency in our patient population. So we went from a lot of patients failing this test, even though clinically they seemed pretty okay, to a much lower false positive rate. And so that's why those numbers are really important.
Sure. Talking about the outlook and the perspective, right? Where do you want to take your research from now in a couple of years?
Mm-hmm. Yeah, I think so. I want to continue to work in the two areas that I've been working on. So in this endocrinology area, there's a continued need for additional thresholds and cutoffs that are truly based on good evidence. And then, so there's a lot of things that I want to do with kind of the other area. So in identifying, these are called pre-analytical variables. So identifying the best ways to transport samples, to store them, to collect them. And so there's a lot of things that we deal with in that. And there's really just an endless list of things that we need to consider. So that is where I see kind of an ongoing growth. And then lastly, I've started collaborating with a handful of diagnostic companies that are newer to the market and need clinical collaborators to actually run patient samples. So for example, in the US, we need FDA clearance before we're able to sell and market some of these tests. And so in order to have FDA approval, they need to run a bunch of patient samples. And so I have a couple of projects collaborating with industry partners like that. And I hope to do more of that in the future as well.
Good, thank you so much for this information. Apart from that, what we already spoken about, do you think there are any other important information that you would like our viewers to know about?
Yeah. So I think the last thing I want to mention is, so back to my background, I, was like able to kind of stumble into clinical chemistry. I was really fortunate to find this as a kind of a clinical field that I could work in with a PhD. And so I do want to share a little bit more information about those career choices, because I think there's definitely a likelihood that other PhDs will be seeing this or PhD candidates. So there are 3 paths for a PhD in the clinical laboratory. One is in clinical chemistry. One is, which I've described. One is clinical microbiology, which is kind of what I do still in a, kind of a clinical laboratory setting, but looking for microorganisms. And then lastly, molecular genetics. So this would be all of the genetic testing, which is really just exploding. And we need people to run those laboratories and make sure that the tests are done correctly and that the results are easy to interpret. So those are 3 fields I would highly encourage anybody with a PhD that wants to get into more of a clinical field to look into.
Good, thank you so much for this information and sharing your experiences. It was a pleasure to have you with us today.
Thank you so much. I appreciate the opportunity.